This video will cover information related to prokaryotes. This video will go through the understanding, application, and skills for prokaryotes in subtopic 1.2 within the cell biology unit. So there are two main types of organisms here on Earth, and they can be grouped according to their cell type. Cells can be classified as a eukaryotic cell or a prokaryotic cell. Eukaryotic cells contain a nucleus that will hold the DNA for the cell. Prokaryote cells do not contain a nucleus, but do contain DNA that is not stored within a nucleus. Prokaryotic cells can also be thought of or considered bacteria, or also go by the name bacteria. Prokaryotes are single-celled organisms that lack membrane-bound nucleus and organelles. They're the first organism to evolve on Earth and have the simplest structure. Prokaryotes are also the smallest organisms here on Earth, and they range in size between 1 micrometer to 2 micrometers. Bacteria, or prokaryotes, do have a lot of negative effects, so they are responsible for many diseases. So infectious bacteria and other disease-causing agents, such as viruses or funguses or other microorganisms, are known as pathogens, or they can have pathogenic effects. Bacteria can also infect livestock and crops and therefore threaten our primary food source. So there are a lot of recalls, food recalls, because of E. coli contamination or Listeria contamination, both examples of bacteria. And so something to think about, that it might be considered negative for the infected species. There are a lot of negative health consequences to an unwanted bacterial infection, but not so for the bacteria. The bacteria get a warm place to live and access to all kinds of nutrients. It may also not be a negative thing for the predators of the infected species. There are positive effects of prokaryotes. We do use bacteria a lot for our own purposes. Prokaryotes are also important within ecosystems. So they can act as decomposers and they can help cycle nutrients. Bacteria do reside in the intestines of animals and can aid in digestion. So we have a lot of bacteria in our intestinal tract that will help with the digestion of our food as well. And will also produce some nutrients for us, such as some vitamins. Bacteria are also used in commercial areas, such as making yogurt, cheese, and chocolate. Chocolate is made using yeast and bacteria to ferment those cacao beans. Bacteria can also produce antibiotics. So, for example, the common class of antibiotics, the tetracyclines, were discovered from soil bacteria, and since then many different types of tetracyclines have been produced or synthesized. So, with characteristics of bacteria, they are single-celled prokaryotes. And a bacterium now, its chromosome, is going to be a single loop of DNA that is found in a region referred to as the nucleoid. So that chromosome is going to be the genetic material that contains the genes of that bacteria. Is really just a single loop of DNA. The ribosomes that will help make the protein that the cell will need are found scattered throughout the cytoplasm. And bacteria do reproduce asexually by a process referred to as binary fission. So the diagram here goes through some of the common structures that you'd find in a bacterium, and we'll run through what all these structures are. So some of the common characteristics of bacteria is that they often have one or more flagella for movement, and small hair-like structures called villi. The flagella are basically like a tail-like structure that allows for movement. So the flagella will move back and forth and allow that bacteria to move from one position to another. And bacteria can have small hair-like structures called villi, which will allow for attachments to various surfaces. And how bacteria stick to things. Many bacteria have additional DNA within their cytoplasm found in what's called a plasmid. So a plasmid is that small loop of DNA that will contain a small number of genes. These genes are not essential for cellular functions. So if the bacteria does not have this plasmid, it's not detrimental to its survival. But having this plasmid will often offer some type of advantage to cell survival, such as giving bacteria resistance to antibiotics. So those plasmids can contain genes that will code for an enzyme or some type of protein that might neutralize the effect of an antibiotic. A lot of bacteria will have a cell wall that is composed primarily of a compound referred to as peptidoglycan. So this compound or this molecule is a combination of amino acids and sugars. And is different from plant cell walls in that plant cell walls are composed of cellulose. Some bacteria are surrounded by a sticky capsule, which is an outer layer on the bacteria, which will provide some protection for the cell. So in the prokaryotic cell, the main distinguishing feature between prokaryotes and eukaryotes is the lack of a true nucleus within the prokaryotes. So, as mentioned on previous slides, the DNA of that bacteria or that prokaryote is just going to be found directly within the cytoplasm of the cell. There are a number of features to prokaryotes, such as they will have ribosomes within their cytoplasm to help make those proteins that they need. They do have, as mentioned previously, those tail-like structures, the flagella, as well as the pili, which are those hair-like structures. And then there will be, of course, a plasma membrane and a cell wall. It's important to note that any of these items that appear in that red box are items that you would include in a drawing of a prokaryotic cell. So having a look at the different structures on prokaryotes and their functions as well. So we'll start with the cell wall. The cell wall will provide structural support and protection. And adjacent to the cell wall will be the plasma membrane. The plasma membrane, as with any other cell, is going to control entry and exit of substances. And some of these substances will be moved in and out of the cell in a process referred to as active transport. Active transport is where the cell will use ATP, or energy, to actively move a substance from one side, from inside the cell to outside, or outside to inside. The cytoplasm will contain the enzymes needed for all metabolic processes. In the cytoplasm, there will be ribosomes as well, and these are free-floating in the cytoplasm. They're not attached to any particular structures, and the ribosomes are going to be used for protein synthesis, or they are what will be used to create proteins. It is important to note that the size of the prokaryotic ribosome is important. Prokaryotic cells have what's referred to as a 70S ribosome, while eukaryotes have what's referred to as an 80S ribosome. When referring to ribosomes in prokaryotes and eukaryotes, it is important to make that distinction between the 70S and the 80S. And all it's really referring to is the size of the ribosome. So eukaryotes have a slightly larger ribosome than prokaryotes. Other structures found within prokaryotes include DNA, and it's referred to as being naked DNA. So the DNA is found in that nucleoid region, so wherever you find the DNA is the nucleoid region. It is not going to be surrounded by a nuclear membrane, so it's not going to be found in a nucleus. The DNA is going to be circular, meaning it's literally a circular structure. And what makes the DNA not naked is that it is not associated with any proteins. So it is just the DNA by itself. Some bacteria will also contain a structure referred to as pili. These are those thread-like projections and are going to be usually more numerous than the flagella. And they're not usually involved in movement, but as mentioned in previous slides, they allow bacteria to stick to other cells, and they have a special role as well in that they can be involved in transferring DNA to another cell via a process called conjugation. So literally, if you have two bacteria cells next to each other, they can form they can have these pili projections that will connect these two cells together, and this will allow the transfer of DNA. So if this bacteria cell has a plasmid or contains plasmids, it can allow the plasmids to be transferred from one cell to another. So this cell that was lacking a plasmid now can obtain that plasmid and gain any advantages contained within that plasmid. Another structure found in bacteria can be flagella, which again is just involved with movement. So one of the things you're going to have to be able to do is be able to draw prokaryotic cells. But because prokaryotic structures are small, their internal structures cannot be seen using a light microscope. Light microscopes can only see structures larger than 0.2 micrometers, and given that bacteria cells are very small, it's going to be very, very hard to see any detail or any structures of a prokaryotic cell. So to view structures on a prokaryotic cell, you need to use an electron microscope to see that detail. The electron microscope is a special type of microscope that was developed in Germany in the 1930s. It will allow us to see things in very, very small detail at the nanometer level. And it has done things like reveal the structure of cells and has helped identify various organelles such as ribosomes and lysosomes and plasmic reticulum. It's allowed scientists to see the inside of chloroplasts as well as mitochondria. So, with reproduction in bacteria, most prokaryotes will reproduce asexually. The example of sexual reproduction in bacteria would be that process of conjugation. But if a bacterial cell is going to divide, it's going to reproduce asexually. So there are a few distinguishing characteristics of asexual reproduction versus sexual reproduction. In asexual reproduction, a single individual is going to be the sole parent, whereas in sexual reproduction, you have two parents that have to give rise to an offspring. In asexual reproduction, the single parent will pass all of its genes to an offspring, whereas in sexual reproduction, each parent passes half of its genes to the offspring. In asexual reproduction, the offspring are genetically identical to its parents. In sexual reproduction, the offspring will have a unique combination of genes that it has inherited from both parents. So the result in asexual reproduction is that you end up with a clone, so a genetically identical individual. In sexual reproduction, you end up with greater genetic variation within those offspring. And so offspring are going to be genetically different from siblings and parents. So when prokaryotes or bacteria divide by asexual reproduction, they do this by a process referred to as binary fission. So the parent cell is going to divide into two genetically identical daughter cells. Each daughter cell will receive an exact copy of the genetic material from the parent cell. When genetic material is copied, it is possible to make mistakes. And so when mistakes happen, these are what's referred to as mutations. And so it is possible to give rise to a daughter cell that may not be exactly the same in terms of genetic material. Because bacteria reproduce very quickly, there's an increased rate of mutations, or can be an increased rate at which mutations will occur in reproducing that DNA. And so this can work to help increase the genetic diversity within bacteria. So not all of the bacteria are exact genetic clones of each other. There can be slight variations between them, which can help increase their chances of survival. Bacteria, in that process of bacterial reproduction through binary fission, this process can take place in 4 stages. So there is the initial reproduction signal. So the cell receives a signal, which is internal or external to the cell, that will start the cell division process. Next up, there is the replication of the DNA. So if this blue line represents the original DNA within the cell, the next stage is going to be to make a copy of that DNA, which is represented by the red line. This is copied by a process known as semi-conservative replication, which we will cover in the second unit of the course, and what all that means. Once the DNA has been copied, it needs to be divided up between the 2 cells, and this occurs through the segregation of DNA. So each loop becomes attached to some contractile proteins that will pull the 2 loops of DNA to opposite sides of the cell. And then once the DNA has been separated, then the cytoplasm needs to divide, and the cell membranes and cell wall pinch off from each other. And this is the last step, referred to as cytokinesis. So the cytoplasm will divide, and as the plasma membrane begins to pinch and constrict, the membrane fuses and seals with additional new membrane also being formed. And so you end up with 2 daughter cells, which in theory should be exactly the same. All right, so that's the end of this video for prokaryotes.